Okay, so that's a classic Sonic Youth tune. Surprisingly, a 16-year-old boy can play Sonic Youth. Welcome to Kin 425. This is the first of your asynchronous lectures. I'm Phil Chillibeck. I'm your professor for this course. I'm a big Ottawa Senators fan, so I got a kick out of this story last year. How this kid that was a Toronto Maple Leafs fan wanted to have a Toronto Maple Leafs birthday cake for his 8th birthday. And the bakery made him a Maple Leaf Meats birthday cake instead. So you can see his reaction right there. As an Ottawa Senators fan, I hate the Toronto Maple Leafs, so I got a big kick out of this story. The Maple Leaf Meat Company, in response to this story, gave the kid and his family a bunch of free meat. The Toronto Maple Leafs did not give them any free tickets. So these are some announcements for your course. An important announcement here is that there are no labs for the course. You will see that there is a time slot for the labs. In place of the labs this year, you will be doing group term projects on a group term paper on one of the special topics in the course. So this includes fatigue during exercise, sex differences in response to exercise. This can include pregnancy, exercise during pregnancy, sedentary behavior, exercise in environments with altered temperatures, so hot or cold temperature environments, and exercise in environments with altered atmospheric pressure. So this can include altitude or diving. So I request that you get into groups and you do a term paper on one of those specific topics. The term paper is due at the end of the course. So again, this is in place of your labs. So if you are able to form groups, that would be great, like groups of 3 to 4 students. And in place of the lab sessions that are scheduled, you can work on planning and doing this term paper. There's more details in your course outline, but if you have doubts about your term paper topic that you want to focus on, you can email me and let me know what your planned term paper is, and I can give you some tips on how to revise it or how to do your term paper. So today's class is a review of structural and functional properties in muscle, fiber types, and important components involved in contraction and relaxation. So I've got this slide the first few slides here should be review for you. So the first question here is, what are the contractile proteins? The answer here is actin and myosin. Those are the 2 proteins in muscle fibers that are involved in actual muscle contraction. What are the regulatory proteins? These are troponin and tropomyosin. So these 2 proteins regulate whether contraction will actually occur. So they regulate whether actin and myosin will form cross bridges to allow muscle contraction to happen. And which proteins make up the thin myofilament? This includes actin, troponin, and tropomyosin. So the thick myofilament is your myosin, and then the thin myofilament is actin, and then your regulatory proteins, your troponin and tropomyosin. Again, this is a review based on Kin 225, so it's a fill in the blanks question. During muscle contraction, calcium binds to, the answer should be troponin, causing tropomyosin to be lifted off actin, allowing myosin cross bridges to attach to binding sites on actin. So the first blank there should be troponin. The second blank is tropomyosin. The third blank is actin. The fourth blank is myosin. And the last one is actin. On this diagram, this review has to do with muscle structures. So I've got the labels there: myofilaments, myofibrils, muscle fibers, actin, myosin, and I want you to fill in labels on this diagram. So I'm going to go to a printout here. And unfortunately, my younger son destroyed our printer, so you can see the printout isn't of the highest quality. I'm going to put some labels on here for you. So you've got the whole muscle that I've drawn out here. And if you take a chunk of the whole muscle, you get these cylinders that correspond to the actual muscle fibers. So these are your muscle fibers. And then if you take your muscle fibers and you look at the muscle fibers under a microscope, you can extract individual tubes from the muscle fibers that are referred to as myofibrils. And then within those myofibrils, you'll see these components called myofilaments. And these myofilaments include your contractile proteins. So they include your actin and myosin. So your myosin are the kind of thicker contractile proteins, and then your actin are your thinner contractile proteins. And it's the interaction between actin and myosin, so they form cross bridges that cause muscle to contract. So on this diagram, I have a labeling of the contractile and regulatory proteins. So myosin, actin, tropomyosin, troponin, and also ATP. That's your energy currency for muscle contraction. So to fill in these labels here, this first label here would be myosin. This label here would be your ATP that's associated with your myosin. This label would be your tropomyosin. So that's this structure here. This label would be your troponin. And this final label here would be your actin. So this whole thing here is your actin. On this diagram, again, this is a bit of a review. I want you to label the A band, the I band, H zone, actin, myosin, sarcomere, and Z disc. So again, I'm going to go to a printout here to give you the answers. So on this printout, in this box, we have your thick contractile protein. That would be your myosin. Your thin contractile protein here would be your actin. So they have to form cross bridges together for your muscle to contract. And then up here you have your actin, your myosin, and you have these different bands. Let's see, this one band here, this kind of light in color, is your I band. So the I band actually only contains actin. And then here you have your A band, which is kind of a darker band in these structures. So your darker band contains myosin, but it can also contain actin. So that's your A band. This box here corresponds to, and this is a little bit tricky, this is your H zone. So this is kind of a portion of the, sorry, I kind of messed that up a bit. This is your I band. This corresponds to only actin. This is your H zone. So your I band corresponds to the portion that contains only actin. The H zone is kind of a lighter portion of your A band that contains only actin. So the I band is only actin. The H zone is part of the A band, and it contains, again, it contains only actin, but the actin you can see overlaps with the myosin here. These last boxes here, this box and this box, you can see the arrows are pointing to these structures. These refer to your Z discs. So the Z discs anchor your actin in place. So they're kind of structural proteins that hold your actin in place. And then this box corresponds to this region here that represents your sarcomere. So the definition of a sarcomere is it's your basic contractile unit of a muscle fiber. So the sarcomere is bordered by Z discs, and it contains both actin and myosin. So you can see what happens when muscle contracts. You get kind of a disappearance of your H zone. The I band becomes a little bit shorter. The A band stays the same length. So those are different things that happens during muscle contraction. Okay, this slide just shows a blow up of how myosin actually looks. So myosin actually has two heads, and each head contains proteins that correspond to heavy chains, and then they have two light chains. The type of heavy chain determines the myosin ATPase activity, and that's ultimately going to determine your speed of muscle contraction. So the higher this enzyme activity, the higher your speed of contraction. And the myosin is referred to as the thick myofilament. The thin myofilament is your actin. The thin myofilament is your actin, but also your tropomyosin and troponin as well. So the ATPase that's associated with myosin heavy chain, it catalyzes the breakdown of ATP to ADP and inorganic phosphate. So that's adenosine triphosphate to adenosine diphosphate and phosphate. And the breakdown of this molecule gives you your energy for muscle contraction. So when you break down this molecule, you get a release of energy, and that drives muscle contraction. The higher amount of ATPase that you have, the faster the reaction can occur, and the faster the muscles can form cross bridges. So like a fast twitch type 2X muscle fiber would have a high amount of ATPase and give you a fast speed of contraction. A slow twitch or a type 1 muscle fiber would have a low amount of ATPase and a slower speed of contraction. So fiber types can be classified as type 1, type 2A, or type 2X in humans. And those correspond to slow oxidative, fast oxidative glycolytic, fast glycolytic. And in turn, those correspond to slow twitch, fast twitch A, fast twitch X, or slow, fast fatigue resistance, fast fatigable. Each of these classification systems is based on different measurements. So with regards to the type 1, type 2A, type 2X, it's based on your myosin ATPase activity at your myosin heavy chain. So the type 1 has a low myosin ATPase activity, a slow speed of contraction. The type 2X has a high myosin ATPase activity and a high speed of contraction. The type 2A is intermediate. Slow oxidative, fast oxidative glycolytic, and fast glycolytic, that classification system is based on your enzymes in your muscle. So your oxidative or glycolytic enzymes. The fast glycolytic have a lot of glycolytic enzymes. The slow oxidative have a lot of oxidative enzymes. So the glycolytic enzymes are mainly in your cytoplasm of your muscle fibers. The oxidative enzymes are mainly in the mitochondria of your muscle fibers. Then you've got this other classification system, slow twitch, fast twitch A, fast twitch X, or slow, fast fatigue resistance, fast fatigable. These are based on contractile properties. So if you stimulate a muscle and it contracts fast, it's going to be a fast twitch X. If you repeatedly stimulate a muscle and it has good endurance, it's going to be a slow or slow twitch muscle. Now this slide corresponds to the thin myofilament, which contains actin, troponin, and tropomyosin. So this is a bit of a review from KIN 225, plus a little bit more detail for this course. So your contractile protein there is actin. The actin is made up of two components, so you can refer to it as G-actin, which is the globular actin. So the globular actin refers to each of these little circles. So each of these little pearls refers to globular actin. And then the F-actin refers to filamentous actin. So that's the string of pearls. So filamentous actin would be this whole string of pearls or circles in this diagram, whereas the G-actin is just the individual circles or pearls. This is your tropomyosin, so it lies over your actin. It's a regulatory protein, so it prevents muscle contraction. So it blocks the binding sites on actin for myosin. And then you have tropomyosin, which binds calcium and allows lifting of this tropomyosin off of actin during contraction and allows myosin to bind to the actin. So troponin is made up of a number of different components. So this is beyond KIN 225. So you have first troponin component called troponin T, which binds to the tropomyosin. Then you have troponin I, which stands for inhibitory. It positions the tropomyosin over the binding site on actin. So it prevents muscle contraction by preventing myosin from binding with actin. And the last component of troponin is called troponin C. It stands for calcium, and that's where the calcium binds to troponin. So during muscle contraction, you'll have calcium coming in here, binding to troponin C. That causes the whole troponin to lift the tropomyosin off the actin and allows myosin to form cross bridges with actin to allow you to have muscle contraction. And the whole thing is called the thin myofilament. So the thin myofilament refers to actin, tropomyosin, and troponin. Now, another component of muscle fiber typing is that troponin and tropomyosin exist in fast and slow isoforms, similar to myosin. So myosin, exists in different isoforms like type 1, type 2A, type 2X. Troponin and tropomyosin also exist in fast and slow isoforms. So if you combine the two, you can get quite a high number of different muscle fiber types. Just to backtrack here, the only protein out of all these proteins that I've discussed so far, so actin, myosin, troponin, and tropomyosin, the only protein that doesn't exist in slow and fast isoforms is actin. So actin is the same protein in a fast twitch or slow twitch muscle fiber, whereas troponin, tropomyosin, and myosin, those proteins differ for fast and slow isoforms across different fiber types. These are just additional types of myosin heavy chains. So your myosin heavy chains determine your speed of contraction. So myosin heavy chains correspond to your muscle fiber type. So you have myosin heavy chain 1 corresponding to muscle fiber type 1, myosin heavy chain 2A corresponding to fiber type 2A, myosin heavy chain 2X corresponding to fiber type 2X. You also have neonatal and embryonic myosin heavy chains. Those are seen mostly during development in humans, but you also see these myosin heavy chains and specific fiber types popping up when you have regeneration of fibers that have been, either following disease, or if you're talking about exercise training, these fiber types pop up if you have muscle fibers that are either being formed with exercise training, or if they've been damaged with exercise training and they're being repaired, then you see these myosin heavy chains and these different fiber types pop up. So you see these neonatal and embryonic fiber types or myosin heavy chains being expressed during development, during different disease processes, during different diseases, or if you have a muscle fiber that's been damaged during exercise and is being repaired, or you have new fibers that are being formed during exercise in response to exercise training. Along with the myosin heavy chains that are associated with the myosin, you have these light chains. So again, you have 4 light chains that are associated with each myosin molecule. And these light chains, again, they exist in fast and slow isoforms. So they, again, increase the amount of fiber types that you can have. And these light chains are just thought to regulate actin-myosin interactions. So they can influence whether actin and myosin are going to be fast or slow with their formation of cross bridges. So going back, a few slides, I had, at the bottom here, I had the contractile properties, to define different fiber types. So this review here has to do with contractile properties. So if you deliver a stimulus to a muscle fiber and you get these force time tracings, the question here is which graph represents a slow twitch versus a fast twitch muscle fiber, and why? The top graph represents the fast twitch fiber because it has a fast time to peak tension or peak force. So you can see it reaches peak force quite quickly, whereas this one down here reaches peak force quite slowly. So it takes a greater amount of time to reach peak force. The fast twitch fiber has a fast relaxation time, so it comes back down to baseline quite quickly, whereas the slow twitch fiber takes longer to come back down. And the fast twitch fiber also has a higher peak tension, so a higher force development compared to the slow twitch fiber. So those are the 3 characteristics from a force time tracing that you can use to distinguish a fast twitch versus a slow twitch fiber. Now this graph represents force time tracings from repeated muscle contractions. So again, I'm asking you here which graph represents a slow twitch and which represents a fast twitch fiber, and why. So the top graph represents a fast twitch fiber. The bottom graph represents a slow twitch fiber. The fast twitch fiber, you can see it has a greater force development at the start of contraction, but as you repeat contractions, it fatigues quite quickly, whereas the slow twitch fiber is fatigue resistant. It starts off at a slow force development, but with repeated contractions, that force is maintained. So that's characteristic of a slow twitch fiber. It has low force output, but you can maintain the contraction force over repeated contractions better than you can with a fast twitch fiber. So the fast twitch fiber is fatigable, the slow twitch fiber is fatigue resistant. This last graph shows some what are called morphological properties of muscle fibers. So in this diagram, I'm asking you to identify the fast versus slow twitch muscle fibers based on different properties. And I want you to give 3 reasons for distinguishing the fast versus slow twitch fibers. So here I have mitochondria that are labeled. So the mitochondria are where all your reactions for aerobic metabolism occur. Your capillaries are involved in delivery of blood and oxygen to the muscle fibers. So the answer to this question is these darker fibers are your slow twitch fibers, and these lighter colored fibers are your fast twitch fibers. The reasons, the slow twitch fibers have greater mitochondria, so they have a greater capacity for aerobic metabolism. This gives them a greater endurance. And they have a greater amount of capillaries, so they have a greater capacity for oxygen delivery to the muscle fibers. The third reason here in the answer is the color of the muscle fibers. So the slow twitch muscle fibers are darker in color, and that's due to a protein called myoglobin. So I'm just going to spell that out here. So myoglobin. So myoglobin is a protein within the muscle fibers, and it acts as a transporter of oxygen from the capillaries to the mitochondria. So the mitochondria use oxygen in aerobic metabolism to break down your proteins, fats, and carbohydrates. The capillaries deliver oxygen-rich blood to the muscle fibers, and your myoglobin takes the oxygen off the capillaries and delivers it to the mitochondria. So your myoglobin are high in your type 1 or slow twitch muscle fibers, and the myoglobin gives the muscle fibers kind of a red, dark color, whereas the muscle fibers that are low in myoglobin have a lighter colored appearance. And those are your fast twitch fibers. So your slow twitch fibers have high mitochondria, have high capillarization, and have high levels of myoglobin. And your fast twitch fibers have low mitochondria, low capillarization, and low amounts of myoglobin. So the next slide I have here is to identify fast versus slow twitch motor units. So recall from our first review exercise that I gave on the first day that a motor unit is your motor neuron in your spinal cord and all the muscle fibers that is connected to. So in this diagram, we have 2 motor units. This is one motor unit, it's the motor neuron connected to all its muscle fibers, and this is the second motor unit, the motor neuron connected to its 3 muscle fibers. So the question here is to differentiate between the fast twitch versus slow twitch motor units or muscle fibers. The answer here is that this is your fast twitch, this is your slow twitch. So the fast twitch, the motor neurons are larger, and the slow twitch, the motor neurons are smaller. The fast twitch motor neurons are connected to a greater number of muscle fibers. The slow twitch motor neurons are connected to a lower number of muscle fibers. So a fast twitch motor unit has a higher number of muscle fibers. Slow twitch motor unit has a lower number of muscle fibers. And what's not too obvious from this diagram is that the axon of the fast twitch motor neuron is actually thicker than the slow twitch motor neuron. So those are three things that you can use to differentiate between fast twitch versus slow twitch motor units. The size of the motor neuron is larger for fast twitch, smaller for slow twitch. The axon is thicker for fast twitch, thinner for slow twitch, and the number of muscle fibers in a fast twitch motor unit are greater than the number of muscle fibers in a slow twitch motor unit. And the last exercise I have for this lecture is, this has to do with coordinated muscle contraction and all the elements that are involved in muscle contraction. So I wanted you to place all these labels onto this diagram. And again, I'm going to go back to my low quality printout here and try to label these